Hey, I'm not here, so I'm going to do a very quick video just to walk you through the summary that I did for this paper. And I'm going to try and do it in under 3 minutes, which I'm guessing is probably what we have to do in the class. So I thought I'd walk you through some of the things that are key points of the paper. Green fluorescent protein is obviously a protein that glows when it's put under certain light. And it's therefore used by researchers to see where proteins are being expressed in a living organism. So if you attach the protein you're interested in to this green fluorescent protein, it means that you can then see whereabouts in the cell it's being expressed. So it acts as a kind of tag. What's significant about this paper is that it's taken the process one step further. So it's using this process to not only look and see where proteins are being expressed or what locations they are, but also what other proteins they are interacting with, and allowing you to purify and spin them down. So what happens is they create a complex by using antibodies, and in this case they're usually antibodies that are literally anti-GFP antibodies, and they are effectively attached to the complex that you've already got, which is the GFP, the fluorescent protein, and the protein you're interested in. What then happens is that complex is attached to a special thing called dyno beads, which are like a magnetic epoxy beads, and then with that process you use a magnet effectively running these beads through a solution to separate out just the proteins that are associated with the protein that you're looking at. And then that at the end of that process, what you've extracted is then stained with Kumasi blue, and you can look under a mass spectrometer, which allows you to then see what other proteins have been attached to the protein you're interested in. Those are clearly then proteins that interact with the protein you're interested in, and you're starting to form a kind of web image of other proteins associated with that protein. The cool thing is you can then use the same process for those new proteins you've discovered, tack the fluorescent protein onto it, tack the antibodies, run it through again and see what other proteins are associated with that protein. So you're beginning to build up a web of understanding of how the interactions occur. They worked hard to make this process as efficient as possible in various stages, because the quicker this process operates, the less changes you're going to see in molecular structure and interaction, so the more realistic indication of what's going on happens. The reason that they're confident that this worked well was because they use known complexes like AP2R, which is a clathrin associated protein complex, where we already understood the pathways very, very well. So it was great for them to be able to compare what we already knew with the results they were churning out with this new technique. And the new technique was confirming what they already knew from existing information. It was done with a number of other complexes like the ARP2-3 complex and various others as well. The really cool thing about this, there's a couple of factors here that are interesting. The first one is that the speed means you've got good accuracy. But secondly, there's a large number of strains of research cells that already use GFP protein, so that means you've got a wide range of existing cell strains and laboratory strains to work on. But also the most interesting thing is that so far the results they looked at in this paper also show that this works across a multitude of species as well, which gives you a huge opportunity to study multiple species, which will only help understand protein interactions even more. So there you go, in under 4 minutes, but 3 minutes plus change, I've given you a brief summary of this paper.